Okay, so anybody here want to do a quick recap of the last thing we did yesterday? Anybody feeling brave enough? Just the last thing we did yesterday and why we did it. Mm-hmm. All right, and what did we see when we looked at that? Okay, so the last thing we did yesterday was we took our raw read file, completely untouched at that point. We ran it through FastQC. FastQC did some metrics on it. The main one we're concerned with is the fact that there was a lot of low quality read. So you could actually see that a lot of the reads, as it approached towards the end of the read, the quality just started getting terrible. That's a pretty common thing to happen with DNA sequencing, and there's computer programs that'll go through and basically prune off where the read quality starts to fall off. So yesterday we discovered that we had a read quality problem. Anybody want to guess what our next step's going to be? Trimming. So the first thing we're going to trim off is adapters. Anybody here remember why we want to trim off adapters? Yeah, it's artifact. It's artifact. It's something we artificially put in our data, and we want to get it out before we try and analyze something we know is bogus. So here's a more challenging question. Can anybody here think about why we would want to trim off adapters before we trim off low quality sequence? Yeah, so that's one very good answer. Is if you trim off a low quality part of the adapter, you may be left with so little adapter you can't recognize it as such. So trimming off low quality sequence decreases your chance of recognizing an adapter. Can anybody else think of another reason? So that is unlikely to happen because adapters usually show up at the far end. So adapters typically show up at the low quality end. So there's going to be another reason, and it's not obvious now, but it's possibly going to become more clear when we start getting ready to do the quality trimming. All right, so let's go on ahead. Everybody should get logged into the Hoffman cluster. Before you log in, if you're here in person, make sure your computer is hooked into the Collaboratory AP Wi-Fi, so Collaboratory AP 5G or 2G. Make sure you're not hooked into one of the UCLA Wi-Fi, so EduRoam, UCLA Web, or UCLA Wi-Fi. There were quite a few people yesterday who were having their sessions drop because those Wi-Fis are not completely reliable in this room. Oh, the security key is going to be on the wall. So yeah, we used to have much better Wi-Fi in the room through the campus, and then they did a bunch of construction on either side, and I think they must have moved the routers around because now we get really unreliable Wi-Fi in here through the campus. If you use Collaboratory AP, you're connected to that router over there in the corner. That's going to be the best Wi-Fi you can get in here. All right, and it's 9:15 now, which is, I think, a good California 9 o'clock. So let's get rolling with this thing. So, all right, as soon as you get who here has not gotten logged into the cluster yet? Okay, I know you're still getting your stuff ready. As soon as you get logged into the cluster, immediately do QRSH, and then immediately do CD space $ scratch. So you're going to immediately request an interactive node. As soon as you get your interactive session going, you're going to immediately go and get yourself into your Scratch folder. All right. Is anybody having trouble logging in? Oh, you're having trouble logging in? Oh, okay, so it's going to be Hoffman. Get rid of one of those N's at the end. .idre.ucla.edu. All right, now you're just going to give it your password. There you go. Okay, so the first command you're going to do is QRSH. And then as soon as you do that, do you remember how to go into your Scratch folder? CD, and then $ scratch, no spaces, all capital letters. Okay, anybody else having trouble getting the cells connected? Cool. All right, so I'm going to get moving then. And once you get into your Scratch folder, actually go into the Workshop 2 data folder. So that's going to be CD. Well, anybody here know what the command is going to be to go into the Workshop 2 data folder? So yeah, it's actually going to be, so the directory structure is going to be CD and then WS2/Workshop2 data. And when you type that, remember that tab autocomplete is your friend. All right. So when you do QRSH, it's going to take you back into your home folder, no matter where you were before. Yeah. So once you get back your command prompt and you're on interactive node, that's when you're going to connect into your Scratch folder. Yes? You're getting what? Oh, yeah, that's a normal warning message. It's probably just hanging there and waiting. You're just going to wait on it. Okay, turning down the lights a little bit so the screen's a little easier to read. I think somebody said something in chat. Okay. So again, from yesterday, we looked at our quality scores. There we go. We looked at our quality scores and we figured out a few things about them. So just like a lot of other DNA sequencing, as you get further along in the read, so again, remember, x-axis is position in the read, y-axis is quality, and we see that early in the read, quality tends to be very high. Later in the read, quality is dropping off quite significantly. So we just want to prune back any reads that have basically garbage at the end. There we go. For some reason my clicker wasn't working at first. All right, so A couple of other things that FastQC will tell you. I'm not going to go too much into detail on these ones. Why do I keep getting that warning? Yeah. All right, so FastQC will give you a couple of other pieces of information, such as sequence duplication. Anybody here know what a PCR duplicate is? Okay, Yeah, so a lot of times during library preparation, you're doing some rounds of PCR. It's possible during those rounds of PCR that something gets duplicated more than it should, or the same piece gets read more than once. And there's a few different ways to pick this up. Some people use these unique molecular identifiers, and other people will just go by the fact that if your fragment starts and ends in the exact same place, then it's probably, or if your fragment has the same sequence, then it's probably a PCR duplicate. Let me double check. I keep getting an alert message on there. I'm not quite sure why. Okay, people on WebEx, tell me if this one is loud, so the first time I say this, or if it's louder on the second one, which I just said. Okay, thank you. I keep getting an alert saying I need to adjust my mic. Okay, I think that's about as good as I'm going to get it, but I think audio should be loud and clear now. So again, PCR duplicates, you want to detect those. Anybody have a good reason why we want to detect a PCR duplicate? Yeah, it's an artificially overrepresented sequence. So if you're looking for variants, you'll think you saw that same variant twice. So it might be that your program, if it sees a variant once, assumes that it's an error. And if it sees a variant multiple times, then it figures it's probably real. And if you start counting PCR duplicates for that purpose, then you'll see a lot of things that you're seeing multiple times, but due to technical reasons in your experiment, not because it's actually there more. And if you're doing RNA-seq, that can get even more confusing because you're basically counting something twice. You're artificially doubling the expression of something by doing that. If you count a PCR duplicate of it. All right, so FastQC is also going to look for overrepresented short sequences, and a lot of times these overrepresented short sequences tend to come up from basically from adapters or primers or other sequences you put in there artificially. Primers and adapters are the main source. You should see pretty even base frequencies across different cycles. So you see this graph right here. You probably noticed that your ATs and your GCs, they're pretty much even all the way across. You get a little bit of non-randomness at the beginning, otherwise it's essentially flat all the way. This is one of these things that I say, this should be boring. If this graph looks interesting, that's when you start to worry. Just curious out there, a plug for another class, who here would be interested in learning how to write code on your own that does this? So if you take the advanced Python workshop next quarter, our final program is actually writing a Python script that goes through line reads, but generates this graph. So if one day you want to start making your own bioinformatics tools, that's a good place to get started. Advanced Python has a big focus on teaching you how to sort of approach writing a computer program. All right, so a couple of other things it'll tell you. In RNA-seq, you tend to get a lot more noise at the beginning of the read, but then it also tends to level out. So who here does RNA-seq? Okay, got one person. Do you know what you frequently prime to when you do RNA-seq? So a lot of people, well, people are using either random primers, or some people are priming to poly-T, or are using poly-T to prime to poly-A. So typically in RNA-seq, there's a lot of issues with non-random priming. So a lot of times the sequence will begin with sort of uneven letters, which is what you see here. So you see here the graph is not boring. The graph gets a little bit interesting. If you look here, you can see that all these reads start with about the same letters. So that's actually some barcode that got in the beginning. So basically that means that you have adapter contamination. We can get rid of that easily. And with adapter contamination, we get rid of this with Sive. So I mentioned before, Sive is quality score aware. And I'm going to show two different examples. So here it's trying to match, and it sees two mismatches between the expected adapter and the actual sequence, but the mismatches are in low quality bases. So if the mismatches are low quality base calls, it's going to assume that it's just an erroneous base call, and it's going to treat it as an adapter anyway. On the other hand, if there's mismatched high quality bases, it's going to figure that it's not actually looking at an adapter. So essentially, it's just looking at sequence at the end and matching it to known adapter sequences and saying, what's the probability this is an adapter? And to do those probabilities, it's going to look at both the matches and mismatches and how likely it is that the base that was called is the base that was really there. Anybody who knows a little bit of Bayesian statistics can kind of imagine how this system works, because I'm pretty sure that Sive uses a Bayesian method. All right, trimming with Sickle. So Sickle is a program that looks for bad quality sequence, and it uses a sliding window approach. Sliding window is probably the #1 algorithm you're going to come across as a basic DNA sequencing, sequence analysis algorithm. That's taking a window of a set size. So here it's drawn as 8. I think in real life, Sickle uses a window size of generally about 15. But it's taking all the bases in this window, calculating the average quality of those bases, or the average confidence score, and then asking if the average confidence score in that region is greater than the minimum threshold. So let's say that I want the region on average to have a 99% confidence. Typically, you're going to get that early in the read. You're going to drop down below 99% confidence probably as it gets towards the end. So it's going to take a window, it's going to measure it, it's going to move over 1, it's going to take the next window, measure it, move over 1, keep going until it hits a window where the quality is bad, and then it's going to say, okay, I hit a bad quality window, I'm going to actually cut off at the beginning of the window. All right, sometimes you move your window across the read, and you find out within 2 or 3 bases that the entire read is crap. It's guaranteed to happen during a sequencing run. You're going to get certain reads that are just garbage from the get-go. So those reads, you just got to get rid of them entirely. So anybody out there want to take another crack at why you want to get rid of adapters first and then get rid of low-quality sequence? Okay, so this one's a little bit more of a challenging question, but it's entirely possible, although very unlikely, but it is possible that you trim off low-quality sequence from a read, and the read length is still fine. And then you trim off adapter sequence, and all of a sudden the read is now too short to process. So your adapter remover is generally not set up to try and remove read that's too short. The quality trimmer is set up to remove reads that are too short. So why do we need to make sure we get rid of very short reads? Mm-hmm. So a lot of times if you're going through a core, they remove the adapters for you, I've noticed. So a lot of times you're going to get back something that's already adapter free. And some of them I think might even do quality trimming, but they tend not to do quality trimming as much, but they do tend to trim off their own adapters, which saves you from having to find out their list of adapters. So, oh, go for it. Exactly. That is the exact answer. If you try and map a read of length 8 to the genome, it's going to map everywhere, and it's going to make your aligner very unhappy with you. It's also, when you look at the alignments of it, it's going to look like garbage. So typically you want reads of at least length 20, probably even more importantly length 30, because there's a lot of 20 mers that you'll find scattered throughout the genome. Once you're looking at a 30 mer, you're probably going to be pretty unique, assuming you're not looking at a repeat. So for genome assembly, I would not want a read that short. A read that short is not going to be all that useful. For genome assembly, I'm going to want the longest reads possible. That's why a lot of genome assembly now, a lot of genome assembly now has a tendency to start building using PacBio. So PacBio is the platform that gives super, super long reads with low per base accuracy. So they have to model a little bit around the fact that about a little more than 1 in 10 base calls is probably going to be wrong, but they can error model around that. But the important thing with PacBio is it can read several thousand bases in one read, which is super effective for mapping. So I think the approach right now is to generally use PacBio to start assembling the reference genome, and then start using Illumina reads to kind of weed out the little single nucleotide errors. I think so. I'm actually not, I haven't done much in the way of assembly before, so that's not something I'm super familiar with. Typically I'm dealing with genomes that are already pretty well established. Like usually I'm either working with mouse genome or human genome, which are probably two of the best polished genomes out there. All right, fair warning, there's a trap involved here. So paired end reads are frequently going to come to you in two separate files. And just about every program handling paired end reads is going to assume that that the first read in your first file is a mate of the first read in your second file. Second read with second read, third read with third read, and so on. If you're running something that, like a quality score trimmer, that can potentially throw out reads because they're too short, you can end up with this situation. Read 1 and read 4 were removed from paired end 1, probably because they were garbage and ended up too short. So now we have read 2 match with read 1, read 3 match with read 2, 5 with 3, 6 with 4, 7 with 5. What's this going to look like when you align it? Not so much insertions and deletions. You're going to see more stuff that looks like chromosomal translocations. It's going to look like you have more chromosomal translocation than not. Although That won't actually happen, because what's going to happen is your aligner is going to look at the read names, and it's going to say that read 2 has a different read name than read 1, when really both paired ends should have the same, basically should have the same read location on the, they should have the same read identifier, with the exception that one's paired end 1, one's paired end 2. So if somehow you manage to still end up analyzing those reads that are now unmatched, it'll look like a ton of translocations. In reality, what you're going to get is just you're going to get an error from your aligner saying, hey, something's wrong with your files. So the way that SYCL deals with this is if you have this situation where read 1 and read 4 have to be eliminated, but in paired end 2, read 1 and read 4 are still good, SYCL is going to pass them off to a singletons file, which are effectively now single-ended reads. All right, so this is kind of the exercise to see how we're building up a command line for these. So you're going to start off with the directory for sive. You're going to give the command to run sive. The -a is going to be your adapters file. The -q is going to be your quality score scheme. -o is output. And then the thing with nothing in front of it is going to be your input file. So it's going to look a lot like this. So keys to running on the command line, read the manual. Or if you don't have the manual in front of you, at least Google for some instructions on the program. Be careful with spelling. And remember that most software, especially in bioinformatics, is written by different people. And a lot of times it's written by people without a strong background in writing software. So you're going to see some very different behavior between programs. One of my favorite examples is if you use a program that I wrote, chances are it won't let you overwrite existing data. If you try and output to a file that is already there, it'll either not work at all, or it'll tell you, hey, do you really want to overwrite this file? Some other programs and some other people don't build in that kind of safety. They just assume that whatever you're telling it to write to is where you want to write to. And if there's already a file there, you're going to clobber the file. So different programs are going to have very different behaviors. All right, so we should all be in our Workshop 2 folder. And if you ls in your Workshop 2 folder, you should see a folder called Software. So let's descend into the Software folder. Again, the command is going to be cd and then Software. Is anybody having trouble finding that folder? Awesome. All right, so if we cd into software, since we haven't been in this folder before, the first thing we're always going to do is ls and just kind of have a quick look around. And we see a pair of zip files in there. We see sive.zip and sickle.zip. What do we do when we encounter a zip file that we need to deal with? Unzip. The first thing we got to do is unzip it so it's easy to work with. And if you notice, the command I have up here looks a little bit weird. So it's unzip sive.zip, semicolon, unzip sickle.zip. Does anybody know what the semicolon does? Yeah. Putting a semicolon on a line effectively makes it 2 lines on one. Yes? I don't remember if it can take wildcards. That might also work as well. So why would you want to run both commands on the same line? Save time. I think in this case, I ran these commands and I wanted to get a glass of water. So I knew I was going to run both commands. I just set them both up to run, and I went to go get a drink, and then I came back and both tasks were finished. You're going to get another case where you have a bunch of stuff coming out across your terminal. And once that's done, you should ls again so you can see now what your folder looks like. And you should see two new folders that were created in it. You'll see a scythe master and a sickle master. Is there anybody who had trouble creating those? What error are you getting? Oh, okay. Oh, you probably tried to unzip the same thing twice. So this is a case where it's trying to save you from possibly overwriting important files. So it's saying, hey, if I unzip this, I'm about to overwrite a whole bunch of your crap. All right, so anybody have trouble getting these things unzipped? All right, so now that we have them unzipped, let's descend into one of the directories, and let's descend into scythe-master. And then, of course, since we're in a new folder, we're going to ls first so we can see what's there. And when we get in there, we see a couple of interesting files. We see something called license, probably a bunch of legal mumbo jumbo we don't care about. We see something called makefile. That's kind of interesting. We see paper, okay, whatever that is. We see something ending in .pl, so probably a Perl script. Readme.md, so that is a marked up text file that has instructions in it. If you see readme.txt or readme.md, you're pretty much always looking at something that's going to be a basic set of instructions. We see a file called Selecta adapters and TrueSeq adapters. So a couple of batteries included adapter sequence references. We also see a file called src for source, or actually a folder called src, and a folder called testing. All right, Trevor, I'm going to ask you this one directly because I'm pretty sure you can answer it. If you go into a folder and you see something called src, and you also see something called makefile, it's probably a C program. And more importantly, what's it telling you? Yes. And even more important than that, what is the makefile? It's compiling instructions. Yeah, you can go simple on this one. Yeah. So if you go into a folder and you see that there's a directory called source, src, and a file called makefile, that's basically saying that it's a C or a C++ program. It has to be compiled by the user. And because there's a makefile, it's saying that basically compiler instructions are already included and can be run automatically. So you don't have to know really how to run a compiler. You just have to know how to run a makefile. Fortunately, running a makefile couldn't possibly be easier. You just have to be in the directory and type make and hit enter. And when you do that, you're going to see a couple things pop up on your screen. And then once you get back your command prompt, you're going to do cd and then /sickle master. So you're basically going to go up a level and then down into the other directory all in one line. Once you've done that, you're going to ls to have a look around this new folder you're in, and you're going to quickly notice that it also has a makefile and a source directory. We know how to deal with this now. So you're just going to type make, hit enter, and when you're done with this one, hit ls, and you'll see that there's actually a bunch of new files that got generated. I'll give everybody a minute or two to get that completed. Also, as a side, oh, yes? I haven't seen that error message before, but I have a suspicion I know what it's saying. Oh, you type makefile. Yep. Even simpler. Also, congratulations, you finally found. I've been doing this class long enough that it's not typical that I get an error message I haven't heard before. Let me give you a slide number real quick. All right, slide 125. Yeah, we actually don't get to the ending slides. I have those there as extra in case anybody wants to see. But did you already do the? Oh, okay, yeah. So you're gonna. Okay, yeah. You go into each directory, type make, and then enter. And then you're going to go into sickle master. So you'll do cd, and then .. /yeah. And then, yep. There you go. Yes? Okay, so the make command, the make files have directions for the compiler on how it should properly compile the program. When you type make, that's actually telling the program that basically runs a compiler to look for a make file in the folder, look for directions in the make file, and then start building out the code, start compiling the code from those directions. So this is essentially doing an installation. The program we're running here is actually our C language compiler. Right. We haven't run either program yet. We've run the C compiler so that each program is going to be ready to run. Mm-hmm. Okay, so here's a quick question. If we didn't have sickle or scythe on there, how would we get them? We download them, yeah. In this case, we'd probably clone it from GitHub. I'm pretty sure these files originate on GitHub, especially because one of them's called master, which is kind of a GitHub type of thing. So if we didn't have them on here, we would actually just be able to download them. And once you download it, you can again run make, and it'll do the thing for you. All right, we're going to do cd and then .. /.. What's that going to do? It's going to take us up 2 levels. So it's going to take us out of the sickle or scythe master, whichever one we happen to be in. It'll take us out of the software folder, and it'll take us back into our main workshop 2 data folder. So from our workshop 2 data folder, we're going to actually call the sickle program, and we're going to call it by doing /software/sickle-master/sickle. This is a really good case to use the tab auto-completion. Yes? So you can totally do that. In this case, we're going back to where our data is. So we're going to run stuff from where our data is, but in this case, we're actually just calling sickle really quickly to make sure it runs. And why we're doing this is going to become obvious in a minute. This makes it easier a little bit later on when you want to hit the up arrow and get back this command to run sickle for real. Because right now we're not actually going to run sickle. So if you run sickle, you should get something that looks a lot like this. This is a very simple set of super basic instructions on how to run sickle. This is a really common thing you'll see in computer programs. If you run a program without options and it needs options, it'll give you some basic instructions or at least list the possible options you can use to run it. We're just going to run sickle. We'll deal with scythe in a bit. So the other thing that running this tells us is if we get back this output of instructions, number one, it'll give us some basic instructions if we haven't run the program in a while. More importantly, you only get this output if the program was successfully compiled. So if the program, for some reason, isn't runnable, you'll actually get an error message saying it couldn't run. All right, so the minimum information scythe needs. Scythe is the one that's going to remove adapters. The minimum amount of information it's going to need, obviously you need to call scythe as a command. It needs to know the adapters. It needs to know where to output the data, and it needs to know the sequence file it's going to work on. So the minimum amount it needs is adapter file is data. It needs input data from your sequence, and it needs to know where to output the result. And if you're not using kind of the standard Sanger quality scheme, then you need to tell it Sanger. Although it's always better to be explicit and say Sanger. So if you look up at the top here, you'll notice that we're using that exact same command before. So how do we get back that /software/scythe master/scythe? Yeah, you can get back the last command with the up arrow. Oh, actually, we just ran sickle. So first one we're going to do is scythe. So in this case, you will need to type it out. It's going to be ./software/scythe-master/scythe. And you were asking why we were running this from our main folder instead of the software folder. So the reason why we want to run it from our data folder is if we run it from our data folder, we have to put a longer path in for scythe. But we can put in a shorter path for the adapters file and for our data file and for our output file. Because all these things are in the same directory, so I don't need to give a longer path. This one may take a few minutes to run, but once it's over, you'll see an output that looks like this, and I'll give everybody a minute or two to get the command typed out. If you run into trouble, throw up a hand, and I'll see if I can help you out. Yes? Are you doing .software or ./software? All right, let's see what's going on. Ah, so you're still in your sickle home. You're still in your sickle folder. Okay. Mm-hmm. Looks right to me. Mm-hmm. Ah, so let me have a quick look here.
Maybe I didn't launch. Does that mean I didn't launch the master? Or run the master command? Yeah.
Wondering if it was not unzipped.
Yeah. So if I go into that,
Okay, so that.
to that directory.
Yeah, that wasn't okay.
So I need to do mate. Okay. And now, I thought I'd seen this. I'd seen this previously.
There you go. No problem. What do you mean? Oh no, once it's compiled, you have all the executable files. It's going to create the new files. They'll be there permanently. Yes. So CD and then dot dot. Okay, so yeah, this is your main data folder. So this is where you want to run things from. /software. Hit tab. Yep. So first run it without any options. Okay, so when you get the instruction, you know it's working. So hit the up arrow now. And now you can start putting in the options. I see. And then the other thing is you need to give it its so that's data going in, so that's your reads. That's going to be data going out, so that's your cleaned up reads. That's the quality score scheme. That's your adapters file, and then that's the command to run the program. So just hit enter. don't know why it's saying that file is unfound. I see that file. Oh, never mind, it is there. Okay, I think you had PEL instead of PE1.
Oh, it's PE1.
Yeah.
Yeah, I did have PEL.
Yeah, that's most of the typos I can pick up really quickly, but the L and the 1 look so similar that I don't usually notice that one. Yes. Mm-hmm.
But then just these parameters with these exact values.
It's not going to be, let's see, it's not going to be with those ones actually. Look for the command line, so scroll down. Keep scrolling. Keep scrolling. Scroll up. You see the command up at the top there? That's the actual command, and then the output's below it. Oh, that's because you're actually looking at the command line on the wrong slide. So the command you want to run is actually there. Mm-hmm. Mm-hmm. Oh, the adapter sequences? If you need to remove adapters yourself, whomever did the sequencing for you should be able to give you an adapter sequence file. Or at least they should be able to tell you what adapter sequences, what adapters they used. And any company that provides adapters is going to provide adapter sequences too. All right, so I think most people have gotten this to run. You'll notice when you run it, you get an output that basically has a bunch of zeros in it. This is telling you where it found contamination of adapter. So, for example, TrueSeq adapter 1 showed up. We also saw adapter 13 show up. So we got a little bit of adapter contamination. Fortunately, the program found it and definitely removed it. And somebody's sending messages on the chat. Okay, so a question came up in chat, and the question was, in the command format, does the out file mean the file after the in file is processed by the algorithm, in this case, Scythe? Yeah, the out file, or output file, that's where the data is going to be put after it's processed. So the input file is going to have reads with adapters still attached. The output file should have the reads with the adapters removed. Okay, and somebody talking about a delay in CyberDuck. So CyberDuck does not automatically update the file, the directory structure. If you look at CyberDuck, you should notice that there's a refresh button. Anytime you expect there should be a new file there that you don't see, hit the refresh button, and then CyberDuck will automatically reconnect and check the directory structure again. But CyberDuck does not maintain a sort of live listing of the files. All right, so we just ran this for the paired N1 file. Anybody want to guess what the next step is? We're going to run this for the paired N2 file. Beware of the steamroller. So I call this occasionally steamrolling, clobbering, overwriting, whatever you call it, it's generally not something you want to do. So always double check your output targets. A lot of stuff you run on the command line has absolutely no problem overwriting stuff that you don't want it to overwrite. And the reason I say that is we pretty much have the command we already want to run on paired N2. So not surprisingly, you want your paired N1 and paired N2 commands to look just about identical. What's going to be ideally the only difference between your paired N1 and paired N2 commands? The input and output. And what's going to be the only difference between those? The PE1 becomes PE2. So we're just going to take the output file here, and so use the up arrow. Don't try and rewrite this command. Just use the up arrow and the back arrow and backspace and some other buttons. And you're just going to change human001 underscore PE1.fastq to human001 underscore PE2.fastq. So just delete the 1 and replace it with a 2. Likewise here, for the output, delete the 1 in PE1 and replace it with a 2. And if your first command worked, then this command should work as well. What happens if you accidentally forget to change the PE1 in your output file? That's where the steamroller happens. You will accidentally overwrite your PE1 file with your PE2 data. So I'll give everybody a minute or two for that to work. This should run a lot faster because everybody should already have the working command. They just need to make a slight modification to it. Scythe, I believe, is one that, yeah, Scythe is one that tends to steamroller. Yes? So you remember how I said that occasionally you can read in either some of your beginning adapter or some of your adapter at the other end if you read too far? That's adapter contamination. Basically, if you look at some of your reads, you'll see sequence in there that you can attribute to the adapters and not to your source DNA. Does that make sense? Okay. So another idea is if I were to sequence, if I were to do an exome sequence on you, I would probably draw some of your blood. I'd extract the DNA from it. I'd fragment the DNA into sort of a predictable size, probably about 400 bases. I then need to stick adapters on either end of the fragments, right? And that's so that I can run it through the sequencer. And the adapters have the priming sites to start sequencing, right? But I don't start reading right at the beginning of the DNA that came from you. I start reading inside the adapter. And if I read all the way through to the opposite end, I might actually read the other adapter too, right? If I read all the way from one end to the other, I'm going to potentially read adapter in there, right? But the adapter isn't DNA sequence from you. It's basically garbage, right? So that's adapter contamination. If I see adapter in the kind of output file or in my DNA sequence, that's adapter contamination, and I need to get rid of it. It can theoretically happen on both ends. Yeah. It means there's probably more contamination, and that might be a function of the fact that maybe you only used adapter 13 for this library. All right. Once this is done, let's see, it's 10:11, so let's do a quick 10-minute break. Actually, let's do 9-minute break so everybody get back here at about 10:20. And I will be here to answer any questions. And I'm actually going to recharge my PowerPoint clicker because it's saying that it's low battery. Yep. Yeah, don't forget to sign in if you haven't. Mm-hmm. Don't worry too much about interpreting those numbers. If you want to know some of the details of how the adapter contamination looked, that's what these numbers will tell you. Usually we're not too worried about that because, yeah, I might want to see what adapters came up, but as a curiosity, I'm more interested in just getting rid of them. So, yeah, the main thing to worry about is if you're expecting a lot of adapter contamination and you see all those numbers come up as zeros, that's when you might want to worry a little bit because that means that it wasn't finding adapter. So for example, if I try and do an adapter removal and like I use the Nextera adapter set for my sequencing, but for some reason I gave it the TrueSeq adapters as the reference adapters. So I basically tried to remove the wrong adapters. Then I'm going to see weird stuff like all zeros. Oh, don't worry about those. You're probably going to have some. So when you think about it, you're giving it a set of adapters, but those aren't. You didn't necessarily use every adapter in the set. So you're going to have a lot of adapters that weren't contaminating because you never used them. So yeah, so if you see an adapter that wasn't contaminating, that's fine, especially if you didn't use it. Like I said, when you worry is if you're expecting to have adapter contamination, and you find that all the adapters that there was no adapter you basically find that you couldn't actually see any adapter contamination. If they're all zeros, that's a good case to start worrying. Then you want to double check and make sure that you gave it the right adapters to look for. Because if you have it search for adapter contamination, but you gave it the wrong adapters to look for, then it's obviously not going to find any, right? I wouldn't say just if there's a positive number somewhere. If you see all zeros in a 1, you might have just seen something by random chance. If you see, but if you're expecting adapter contamination and you see, for example, this, we know that we were finding pretty significant numbers of adapters in there, right? Like I see some hundreds, some 90s, that kind of thing. So we were finding the adapters we were expecting. So that means that basically the adapter removal was successful and found adapters to remove. Yeah. And we weren't finding everything. Like, for example, this one was barely used at all. In fact, these couple might have been a spurious find. This one we definitely found a lot of. Mm-hmm. Yes? Those are going to be all the same. What's actually interesting about those is the ones that directly interact with the flow cell, you'll pretty much never see those as contamination. The reason for that is typically the ones that interact with the flow cell are going to be bound to the flow cell during the run. Right. So the primer's going to bind somewhere downstream of the flow cell interacting area, and it's going to read through. And I guarantee you the primer has to read through your barcode because you need to get the sequence of that barcode to properly demultiplex. Right. Although the barcode is going to be part of the adapter. So the barcode is generally kept a little bit separately from the rest of the sequence. So if you have barcode showing up in your actual read, that's adapter contamination. You need to get rid of it. Yeah, there's usually either a separate file or at least a separate field for the index, which is the barcode. The barcode itself should not show up in the FASTQ's DNA line. It it should be trimmed out because that is basically artifact. It's it's stuff that you put in there. Exactly. Yes. Yeah. If I'm using some if I'm using some homemade adapters, you're pretty much never going to use homemade adapters. I mean, I work at a company where we do, we make some of the stuff people use in labs, so we may or may not be making our own adapters. So so in my case, I might actually have like a custom file of adapters that I'm using. But generally, if you're working in a lab out here, you'll be using some set of commercial adapters, and the commercial adapters all should have files with their adapter sequences readily available. If for some reason a company doesn't want to make their adapter sequences readily available, don't use them. hmm The accuracy of this program. So for that, you pretty much have to dig into the program and see how the code works. Yeah, a lot of times in terms of vetting programs, a good way to do that is, if you can't go into the program yourself and see how it's working, or even if you can, but you realize that the statistical methods it uses don't make sense to you. A lot of these programs have papers that go with them. So you can look at the papers and that'll give you a good idea of what's going on. Or if you can't, or you can also look and see where they're cited. Typically, if it's a good program that a lot of people are using, it should be getting cited in a fair number of papers and pipelines. So if you look at a bunch of sequencing-related papers and they're all saying that they're doing their adapter removal with Scythe, Scythe is probably a pretty good program for doing adapter removal. And the added bonus is, if something turns out to be wrong with Scythe, like it was inducing some kind of bias, number one, nobody's going to fault you too much for it because you're using the same thing everybody else is using. Number two, somebody's probably going to pick it up fairly quickly. And number three, somebody's going to probably come out with a fix for it very quickly. Yes? Mhm. For which one? I'm not super familiar with that one, but generally they should tell you what their FASTQ files have gone through, if anything. So there should be a way to find out how a FASTQ was processed. Is this a file that was made that was directly off the machine, or did they do some cleanup on it? And if they did some trimming and adapter removal on it, they should be able to tell you which version of which program they used and with what settings. If they're giving you a FASTQ and they can't tell you sort of its provenance and what kind of processing it's been through, ask yourself if you really want to use that data. Because if they can't account for it, then you're basically using a set of data that somebody else generated that they can't account for what it's been through. Sort of analogous to my example before with people sneezing and stuff. If you can't say for certain that that their lab tech's last cat video he watched isn't part of the data, don't use it. Okay, checking the, all right, before we get started, I'm checking the comments in the chat really quick. Somebody said the steamroller function works well, so I'm guessing somebody forgot to change a 1 to a 2. I'm slightly disappointed that Yi did not respond back with cat video being in data is actually a plus. All right, so we should have run this on paired N1 and paired N2. So we should have basically adapter-free sequence now, or at least as adapter-free as we can reasonably get it. All right, sickle. Sickle is a little bit more complicated. And I should point out, when you see something formatted like this, these are stand-ins. So you're not actually going to run, these may or may not be the actual things you'll put on the command line. A lot of them probably won't be. You know, this is just kind of a generic name for an input file. This is a generic name for another input file. This is Sanger, so this will actually be what you put in. Generic name for an output file, generic name for an output file, generic name for an output file. And these are some, obviously these are real integers, so these might be values you put in. Although we tend to leave these off because I usually run it with the default settings. I think somebody said something in chat. Okay, and Yi's saying, did I hear my name or cat? And the answer is you heard both. I said, I'm surprised you didn't say that, I'm surprised you didn't comment that having cat videos in the data would be a plus. All right, so again, these, when you see these boxes, this is just kind of a generic command line. For the actual command you want to run, look at the screen cap I have of a terminal session. That's really what's going to give you what you really want to run. It'll also tell you what to kind of expect for an output. All right, so just like a lot of other programs, this one we can kind of break it down into what the different parts of the command line mean. So obviously you have to give it a path to the program. Sickle is the command to invoke the program. The first thing you're going to have to tell it is if it's running in paired end mode or single end mode. So for paired end mode, it's called PE. Anybody want to guess what you type there if you're running it in single end mode? SE, yes. So it kind of helps to figure out the logic the person used when they're coming up with their command lines, because it helps you remember how to run the program later on. So -F, this is going to be our input file. Kind of makes sense why they call it -F, right? -R, that's going to be our input file for the other paired end. Can anybody guess why one of them is called -F and one of them is called -R? Forward and reverse. All right. We have -T, that's Sanger. I think they just call it that because they were running low on letters. -O, that's going to be our output file. Kind of makes sense why you call it -O. -P, that's going to be our paired end 2 output file. Anybody know why they call it -P? Because it comes after O. -S, that's going to be our singletons output. So what's our singletons output again? Yes, these are for reads that lost their paired end mate because their paired end mate was basically all garbage. And you're pretty much guaranteed to get a few of those. All right, -Q, that is our minimum quality threshold. So when we run this, we're actually going to run it with a minimum quality threshold of 20. Although we can actually leave it off if we're using a minimum quality threshold of 20. What? That's the Fred score. So what does a minimum quality threshold of 20 mean? 99% confidence on average in the reads in the window. And then J or dash L, that's going to be a required length. We're also going to leave that as 20. You're going to notice when I run this as an actual command, I leave both of those values off. Why can I leave both of those values off? They're default values. So it's good to know what the default values are. If you're running something not on default values, it's always good to kind of ask yourself why. Because a lot of times when people design these programs, they design them with defaults that a lot of people are using. If you're running not on default values, there's nothing wrong with it inherently, unless you don't know why you're running not on default values. Although if you're running on default values, you should also make sure you know why you're running on the default values. All right, so we're going to run sickle now. So this is the actual command you want to run. You'll probably notice a few patterns in how we're doing things. Every time we create a new file, we're incrementing this number at the start. So we're inputting these 1_sci, and we're outputting files that start with 2_sic. And I'll give everybody a minute or two to get this typed in, because I know it's a freakishly long command. All right, who's gotten the command to run so far? Okay, I'm seeing a lot of hands up. Anybody getting error messages back? Cool. Oh, you got an error message? What's your error message? I don't know how you got there, but I have a rough idea of what got you there, sort of. I suspect that somehow you Actually, no, I'm not. I have no idea how it got there. Can you steal your seat for a second? Thank you. So I'm not sure why, but you see how that file has a 0 there? That's an empty file.
Oh. Yeah, absolutely.
So those were created when you ran sickle because it stopped when it ran out of read in Paradigm 1. The problem is since Paradigm 1 was empty, it ran out of read when it started. So when it disregarded the rest, it basically created a bunch of empty files. We'll overwrite those in a minute with actual data. So I'm actually just rerunning your command to create those files. It might have gotten interrupted during the let's see. Yeah, I see no reason why that shouldn't have worked. Okay. It's that typo that I can never see easily. So when you ran this, instead of PE1, you had PEL as the output file. And yeah, it's like it's 1 pixel difference. So unless I look really carefully, I can't see it. Okay, this should work. Yep. Mm-hmm. Yes. So that probably means you have a typo. So let's, can you turn up your screen brightness a little bit? Actually, can you turn up your screen brightness a lot? Thank you. Sorry, it becomes like impossible to see from an angle if your screen is super dim. All right, so hit the up arrow. Let's see the command you ran. All right. All right, let's see what you have in there. Okay, so you notice how you use dashes instead of underscores? So you have two options. You can either move those files, so you can use the MV command to basically change their name, or you can just correct the name when you type the command and have it go to the file with a dash instead of the file with an underscore. I leave that to you. Yeah. So MV and then MV and then the current file name. Yeah, definitely use tab autocomplete. So yeah, so I'll show you how the command's going to look. So it's going to be that. So you'll do the analogous thing for PE2. So you're working ahead? Yeah, you're working way ahead. Yeah, that's the alignment step. We'll get to that in a bit. Did you already run sickle and scythe? Okay, so yeah, you're actually way ahead. So what you did was actually right. I'm going to basically walk everybody into that error. So yeah, just sit tight for a minute and everybody will catch up with where you are. All right, so I think everybody's caught up now. Yeah, again, on the command line, the command line doesn't really try and correct errors for you. The closest it gets is tab autocompletion. So things like confusing a 1 for an L, and for whatever reason, Putty really likes using a font that you can't tell a lowercase l and a 1 apart. They're like 1 pixel difference. If you're on Mac, you have the option to really easily set the font in your terminal. I highly recommend everybody check out a font called Source Code Pro. That's a font that's designed so that no two characters look similar. The 1 and the L and the uppercase I all look very distinct from one another. And I think in Putty you can probably also set your, there's also probably a way to set your terminal font. I just don't happen to know it. All right, so once you're done with this, hit LS and you can see all the new files you made. So where are we at in this pipeline? We ran our cleanup. So the next thing we want to do is we want to see results. How are we going to see results? Yeah? Oh, are you stretching or? Okay. FastQC. The same thing that told us our reads were bad, we're going to use that to see if our reads are now good. So who here remembers how to run FastQC? And what are we going to run fastqc on? So anybody want to say what file we're going to run fastqc on? Yeah. So for the sake of time, we're just going to run it on paired n1. So the file we'll run it on is going to be 2_sci, or I'm sorry, 2_sick_human001_pe1.fastq. So we're basically rerunning our quality analysis program on some files on our data that's been cleaned. Once you get that, feel free to connect with CyberDuck and download the new HTML file you generated. Keep in mind that if CyberDuck is connected already, you'll need to refresh to see the new files you generated. It won't automatically refresh the directory. Yes? You moved the directory yesterday? Ah. Okay, that's weird. I've seen that happen once before. I have no idea how that happens. Are other people seeing that too? Okay, that is weird. Okay, well, if fastqc for some reason disappeared on you, you should be able to remember that there's you'll have to look up the web address for it, but you should be able to just download it quickly. Or actually, if you still have the zip file, okay, if you still have the zip file, just rebuild it from the zip file. So you should just have to unzip it, set permission to set the file as executable again, and then you should be able to run it again. It should just take a minute or two to do. Yeah, I have no idea why that file just massively disappeared on everybody's folder. Yeah, so in this case, go ahead and replace. It'll ask if you want to replace the jar file. Go ahead and replace the jar file. Which one? Oh no, you're unzipping the wrong zip file. You should be unzipping, yeah, that one. Yes? Let me have a quick look. Okay, so the same thing happened with yours. Okay, so yours was another one where they went in, where somehow the file got, your FastQC directory got emptied. So I just rebuilt it for you quickly. I basically just unzipped the file again, which created the directory structure, and then reran FastQC. Did you already rerun FastQC? Okay, so run FastQC on the trim data. Yeah, you can chmod from outside the folder as long as you point to FastQC. So you'll have to point it to it, but you can totally do that. Scroll up. Yeah, in the PowerPoints. This is for genomic data. Microbiome data you're going to handle a little bit differently. Yeah, scroll up, because you're actually way ahead of us. Yeah, there's definitely there is a collaboratory workshop for microbiome data. Weirdly enough, I'm not the one who runs it. It's actually run by somebody from CNSI, but yeah, I haven't been through the workshop, so I don't know how it is. So yeah, that's a little bit closer to where we are. Scroll down. Scroll down a little more. Yeah, scroll down a little more. Let's see. A little more. Okay, yeah, so we're on the next, yeah, scroll down. So yeah, we're rerunning FastQC. Yes. Mm-hmm. Yeah.
Change the permissions, and then it seemed to be okay with that, and then I tried to launch it again, and it says permission denied. This was the right one for the.
So you actually gave execution permission to the directory.
Oh, okay.
So the command,
okay.
yeah. So hit the up arrow, and
So, and then /.
yeah.
Okay.
Yep.
Okay.
That'll fix it.
Thank you.
Mm-hmm. So that looks a lot cleaner, doesn't it?
Yeah.
So that, yeah. Yeah, and now you have a nice, yeah, and now you have a nice green checkbox. So yeah, previously it went kind of like that, where it dropped off a lot at the end. So now it looks really nice because all the sequences that had garbage at the end, you trimmed off the garbage. So yeah, that's what it should look like. All right, so I think everybody got this going. If you haven't already done so, download the HTML file to your computer and open it up in your browser. If your browser disappeared from your computer overnight, that's something I can't help you with. If your browser happened to be Microsoft Internet Explorer, you're better off just leaving it gone. All right, so FastQC report you get back, you should notice that it's going to have a much cleaner looking graph now. So if you remember from before, our graph looked a lot like this, where we had a lot of garbage at the ends. Now our graph looks a lot like this. Our reads look a lot cleaner. All right, so where do we stand at this point? We've rechecked the quality of our not-so-raw reads anymore, and we're now happy with them. So that puts us in a position where we're going to align our reads to the genome. And let's do a quick 5-minute, let's do a quick break for restroom, stretching, whatever else. So let's meet back here at 11 o'clock exactly. And then I'll start talking about alignments and reference genomes. Yes? Mm-hmm. That's telling you should check it out. It means there might be something interesting there. So in this case, one thing it's telling you is okay, yeah, it's telling you to just look at this one because you have a theoretical distribution and an actual distribution, and the actual distribution is a little bit different, but it doesn't really look that different.
Also, I don't understand the concept of search line. Why do you want to see?
Why do you want to see such flat lines? So if you see that every read starts with the same letters, so for example, here you're seeing that at the beginning of here, for example, you're seeing that it's pretty even all the way across. But if you notice that every single read had the same letter or like most of your reads had the same letters at position 9, 10, 11, and 12, that's a cause for concern. Because if you're reading randomly, you should see a pretty random base distribution. If you're seeing what looks like a not completely random base distribution, then you have to worry that something went wrong. Mm-hmm. What's up? Normally you would run paired end 2. For the sake of time in the class, we're only running paired end 1. Yeah. Yeah. If you're running this on your actual data that you wanted to align, and publish and all that stuff, you would run paired end 1 and paired end 2 through quality control. Yeah, so I'm going to show you how to align them in a little bit. Because but you want to run them both through quality control to make sure both paired ends are good quality. Mm-hmm.
So normally, which one I should that's okay? Anything else?
So these all have useful info in them. The main one we're concerned today with is quality. But yeah, definitely. If you're not familiar with FastQC, you should definitely get somewhat familiar with FastQC reports. It'll give you a lot of information on your sequencing. Mm-hmm. Mm-hmm. I don't remember if that exact figure's in there, and it may have changed slightly because of the FastQC versions. Yeah. Yeah, that figure was probably generated 4 or 5 versions ago. Hmm. No. Hmm. Oh, cool. Okay, cool. Are you an MCDB or human genetics? Oh, okay, cool. Nice. Yeah, I saw that. That's my old department, so it was nice to see they're doing a search. Cool. Well, enjoy the symposium. So if what you guys are reacting to is a file popping in CyberDuck you always have to refresh if you expect a new file. Yes? Yeah, so for sequencing selected parts of genomes, usually there's a pretty established protocol for it. So sometimes you can do PCR-based enrichment. Sometimes you can do capture-based enrichment. So capture-based enrichment is what you do for exome. But there are definitely ways to do targeted sequencing. Another really common one is if you, for example, are really interested in the 16S sequence. So a lot of people doing microbiomics are doing 16S sequencing, and they have primers that bind specifically to the common regions of 16S and are targeted to amplify across the variable regions. So is that one of the things you're trying to do? Yeah, I think there's a method to do it. I don't know the method off the top of my head, but I can think of a few ways that it might be done. All right, so it's 11:01, so I'm going to get started with the rest of this thing, and I think we're pretty much in the home stretch now. All right, so who here knows what the difference is between alignment and assembly? These are basically the two things you can do with a genome. Yeah, so alignment, you have reads, and you already have at least a draft of the genome, if not a really well-polished version of the genome. So with alignment, you're taking each read and you're figuring out where in the genome it belongs. With assembly, you're taking a bunch of reads and you're trying to build up a reference genome for whatever you're doing. So if you're starting with a new species that isn't already sequenced, you're probably going to be doing a lot of assembly. If you're working with a model organism or something with a really well-established genome like human, you're probably more likely to be doing an alignment. All right, so question comes up, what to align to? A few things you'll need to do. Pick your species. If you're trying to align mouse sequence to human genome, good luck with that. That being said, I've actually done that before once by accident. Somebody was having me sequence a bunch of cDNA plasmids, and some of the plasmids were actually mouse cDNAs, and I was not told that it was a mixture of mouse and human. That actually worked really well. So anybody want to guess why that worked so well for me? A lot of times I actually found what I was expecting to find in the plasmid. Yes. It just so happens that because they were doing cDNA, cDNAs are the coding sequences, they're really well conserved. So the only mouse cDNAs that weren't aligning to the human genome were mouse cDNAs that did not have a human equivalent. Had I not been doing cDNA, it would have looked very strange. But in general, pick your species. Another thing that I've done before that is also really cool is if I'm looking for just microbial DNA and I want to get rid of any human DNA, I'll align the reads to human and get rid of anything that aligns to human. I'll then align the leftover reads to chimpanzee, and I actually still find some reads aligned to chimpanzee that didn't align to human. And I'm getting a few puzzled looks out there. Why would that be? The human genome is really well polished. It's not perfect. There's some segments that you can still get in chimpanzee, and either the humans have a variant, or it just wasn't a very well analyzed part of the genome, and it aligns in chimpanzee but not human. All right, pick your version. So as I just said, assembly is an imperfect process. Even the human genome, which has been through a lot of drafts and a lot of work, there's still areas that we know we're not very good at. So can anybody give me 2 examples of where we're probably not very good, or even 1 example? Repeat regions. Definitely repeat regions. Repeat regions are terrible. So as an example, if your read length is 400, because you're doing Illumina reads, and you have a 10,000 base repetitive region, can you really tell how long that region is? No. This is where PacBio comes in really handy. So PacBio can read across an entire repetitive region. Now, it gets even worse. If you have a region that is 10,000 bases long and repetitive, and it turns out this repeat is scattered throughout the genome, like let's say an ALU repeat. Who here is familiar with ALU repeats? Oh, nobody knows ALU repeats. ALU is probably the remains of either a transposon or more likely a virus that got into the human genome. Kind of got all over the place in there. And it occasionally even still jumps around and causes some interesting mutations to show up. But you can find ALU and also things like lines and signs, which are these other repeating elements, all over the human genome. And if that repeating element is longer than your genome, or is longer than your typical read length, it's pretty much impossible to place those things, and it's really hard to know exactly what's around them. All right, so again, pick your version. Oh, and another thing where we frequently have trouble is duplicated genes. So sometimes genes get duplicated over time, or occasionally you get like a pseudogene. And if you have a gene on chromosome 1 that's the real gene, and you have a pseudogene of that same gene on chromosome 5, and the sequences are still extremely similar, you may not actually know where you're sequencing. Sometimes you may not even know that there is a pseudogene on chromosome 5, because you always thought that when you see that sequence, it's chromosome 1. So the 2 ways that the human genomes tend to improve, we get better annotation of repeat regions, and we have better understanding of where things like orthologs and pseudogenes are. So if you're doing the human genome, or actually a lot of standard model species, there's 2 main places to get reference genomes from: Genome Reference Consortium and UC Santa Cruz. Anybody here been to UC Santa Cruz? Yeah. It's a good place to get genomes. It's also a good place to get Darth Vader riding on a unicycle playing bagpipes. I think he's still there. So GRC and UCSC, in theory, have the same version of the human genome. They're just different enough to be annoying. So one of these, I think it's GRC. If you're looking at chromosome 18, it'll be identified as 18, just 18. If you're looking at the mitochondrial DNA, it'll be identified as MT. Santa Cruz, if you're looking at chromosome 18, it'll be identified as CHR18. If you're looking at the mitochondrial DNA, it'll be identified as CHRM. You and I, if we're looking at something that says 18 or we're looking at something that says CHR18, we know those are the same thing. Computer programs don't know that. So if somebody does an alignment to the Santa Cruz version and you have a copy of the GRC version handy, it's going to give you all kinds of interesting errors saying that, hey, this genome and this genome, even though they're both human, this genome has a completely different set of chromosomes than this other genome. So that'll get really interesting really quickly. The other thing is they didn't agree before how they were numbering their versions. So the last version of the UCSC version of the genome was HG19. The last version of GRC's human genome was GRC H37. Those two versions are equivalent. Fortunately, they decided to get together and said, okay, UCSC, skip a couple numbers. We're both now coming out with version 38. So the current genome draft, and I think the last genome draft they're going to say because they're kind of considering the genome as somewhat complete now, and they're just going to release patches when they get better information. Current GRC is GRC H38. Current UCSC version is HG38. There is no HG37 from UCSC. And the other thing I'll talk about is decoy genomes. So if I were to draw a tube of blood for myself, extract DNA from it, and go sequence, what species DNA am I going to see? This is not a trick question. Human. If you spend too much time on the internet, you might think like weird lizard person, especially if you have like tinfoil hats around, but I promise human. That said, am I only going to see human? Do I have anybody in this class from Hem-Onc? No? Darn. I know I at least have a couple bacteriologists. Am I going to see only human? So that needle that's going in my arm is going through a whole microbiome. Hopefully not much of a microbiome, because if the tech's not using like alcohol, I'm going to be kind of annoyed. But there's a whole microbiome on my arm. There's also potentially a virome in my blood. So who here knows what Epstein-Barr virus is? Causes mono? Okay, a few people are familiar with that one. Epstein-Barr virus, once you get it, it tends to hang around in your white blood cells. You're going to probably see that if the person has ever gotten Epstein-Barr, you're going to see some Epstein-Barr virus sequence hanging around in their cells. Who here knows what cytomegalovirus is? That one also, I think 30% of people in the US have it. It doesn't really do anything unless you're very sick, in which case it does really bad things. But about 30% of people have it in the US. I happen to know that I don't because I'm a blood donor and they really like CMV negative blood. But you're going to see non-human. So decoy genomes are really good at dealing with that. The other thing is I also mentioned repeat sequences. So decoy genomes are also going to have, basically decoy genomes have a bunch of these sort of fake chromosomes. And their whole purpose is things like Epstein-Barr virus, common commensal bacteria, cytomegalovirus, sequences that we know are present in human, but are either definitely not human or that we can't attribute to anything. They're going to have decoy chromosomes to pick those up. So if I pick up one of these sequences and I don't have anything to align it to, it's either going to come back as unalignable or it's going to come back as aligned to the wrong thing. Ideally, it comes back as unalignable. I'm in trouble if it comes back as aligning to the wrong thing because that's going to mess with my results. If, on the other hand, I have, say, the Epstein-Barr virus sequence in there as basically a known sequence, then any Epstein-Barr virus is presumably going to be aligned there, and I don't have to worry about it crapping up anything else. So decoy genomes can be very useful for things. So if you're aligning to human, especially human has some really good decoy genomes for stuff you expect to find in humans that is either not placeable or not human. All right, so now it's time to do our alignment and mapping, which is pretty much the last thing we're going to go over in this class. So again, the generic command line for this. BWA is our aligner. We're running it in alignment mode, so we have to invoke the program, we have to tell it what mode it's running in. For alignment, what's kind of the least information it needs to know? It needs input, which is going to be reads. What else does it need to know? Yeah, there's only two things that an aligner really needs to know. What's the input data, and what's the reference genome? Who here knows what this thing is? So I know that a lot of people haven't taken Sergey's class where he covers this. Anybody know? Okay, you know that one. Yeah. So by default, BWA outputs its alignment results to standard out. We want it saved to a file, so we're using this thing. And I think in Linux, people typically call this thing a T. People also call it an angle bracket occasionally, but in Linux, especially when you're outputting standard out to a file, which you're doing here, people typically call it T'ing. So we're going to T the output to this output file. The output file? Oh, you can call it whatever you want. The angle bracket? So if you don't tell BWA where to output, it'll output the data to standard out. So you'll actually see it go across your terminal. It won't save it. By doing it that way, you're actually saying, don't put it out to my terminal, redirect what would be going to my terminal to a file. All right, so on the Hoffman cluster, they have this thing called module load, and it basically loads all the associated dependencies for a program. So loading BWA is very easy. Just do module load BWA. You'll hit enter, it'll pretty much immediately give you back your command line, it won't tell you anything. If you don't see anything, that means it worked. We're going to ls one more time just to make sure that what you're seeing in ls looks like what I'm seeing in ls, which should be still your data. And we know we need a reference, so let's do cd into the reference folder and ls. And we should see that we have a chromosome 14 reference. Does everybody see chr14.fa? Cool. All right, so now that we know where our reference is, let's do cd ... and we'll go back to BWA. Or we'll do cd ... and then we'll run BWA, but we'll run it without any commands whatsoever. And when we run BWA without any commands, it's going to give us this typical splash screen saying just some basics on how to run BWA. So once we know we can run BWA, we're going to run this command, which is BWA ALN, so alignment mode. We're going to give it the reference genome, which is reference/chr14.fa. We're going to give it the input file, which is 2_sic_human001_pe1.fastq. So the file we just worked with. And then 3_BWA_human001_pe1. And I have fastq here, but actually ignore that because it should be .sai. It doesn't really matter that much, but the file output is actually going to be SCI format. It's also not actually going to work. Is anybody getting an error message? What's the error message say? Failed to locate the index, which is weird because we saw the, we saw a reference genome there. So what's it failing to locate? Couple options on how to deal with this. So obviously be careful because this T functionality, there is no safety in that. The moment you output something to that moment you use that, you're pretty much going to overwrite the entire file. So a couple options. Breaking computer is always one of them. If you read the error message, it says you're missing an index. As it happens, if you look at this splash screen carefully, you'll realize that it actually talks about you need to index the genome to use the program. Also, if you look at the different BWA commands, there's actually an indexing command. So actually, let's skip these. All right, so for indexing a genome, the only thing that BWA would really need is a genome. So let's do BWA index and then reference /chr14.fa. And as soon as you see this command running, anybody who needs water, stretching, restroom break, or anything, feel free to take that because the command's actually going to take about 3 to 5 minutes to run. It's actually doing something called a Burroughs-Wheeler transformation. Due to time, I probably won't get to how that works in class today, but I do actually have some slides on liner logic. Burroughs-Wheeler transformation is a really cool process, but it's kind of complicated. But essentially what it's doing is it's allowing genome matching to be done with one operation per letter of read, instead of this thing where it's like one operation per letter of read times the length of your genome, which is obviously a lot bigger. If anybody's really curious about that, I can definitely give you some slides so you can understand what's going on with Burroughs-Wheeler. It is really complicated, but it's also a really cool, like, sort of first bioinformatics algorithm. And I'm guessing people here have not run bzip, except maybe you. Have you run bzip? No? Okay. I don't have one. My guideline is generally just be organized. Everybody is going to have kind of their own preference for how they organize things. The only two things I generally say is it should be a somewhat obvious organization. So if somebody else has to go into your directories to find some of your data, they should be able to kind of intuitively figure out where you're keeping stuff. If the files are kind of all over the place, or if the naming conventions don't really tell you, aren't really obvious what's been done to the file, then you should probably see to that. But otherwise, it's kind of whatever makes the most sense to you. All right, anybody's index completed already? Anybody's index still running? Okay, like I said, it takes about 3 to 5 minutes. No, you only have to run the indexing once. So that's actually what's really cool about BWA. The indexing is a very computationally intensive process, and we're just running this on chromosome 14. If we were running an indexing on the entire human genome, the indexing would probably take about 8 hours. But the cool thing with the indexed genome is, if I've indexed the genome and you start working on the same genome, I can just give you a copy of the indexed genome, and it takes as long as it takes to copy it. The other thing is the genome is indexed every time you use it after that. So we're actually going to see that we have about 500,000 reads to align, and the alignment is going to take a couple seconds, which normally you'd expect aligning 500,000 reads to take a bit longer than that. And the reason for that is we kind of front-end loaded the process. We did a lot of work at the beginning, and now every time we run it, we've gotten a lot of work that's already done. All right, whose indexing has completed? All right, I'm going to start talking again because I think everybody else should complete in the next couple seconds. All right, so as you noticed, it went through, did its indexing, gives you a little bit of information about how the indexing happened. I'm not going to go too much into this. I can definitely, if you have questions about it, I can definitely give you the slides on how BWA works under the hood. In terms of, oh, here we go. And it'll say it even took 304 seconds, so it took a little over 5 minutes. So for alignment and mapping, we have that same command as before. And again, the original one, I keep forgetting to fix the typo in the slide. It should be a .sai file that you're outputting to, not a .fastq. So on the command line, what do the file extensions do? Do they do anything for the computer? Computer doesn't actually care. So if you call it fastq, the computer doesn't actually care. But it's useful for humans because it's going to tell a human what kind of file you're looking at. It should be .sci instead of .fastq for the output file. So yeah, it was just in the original command. I keep forgetting to change that one because you run it and then you immediately get an error. But the command should look like this. If you use the up arrow, you can basically just change it really quickly. All right, so we're going to run the same process for PE1 and PE2. Obviously, be careful not to overwrite the PE1 sci file with PE2 sci. Make sure that they're output to their own files. Yes? No, you want the output of this process is actually going to be sci. So it's going to be a sequence alignment index, I think. It's basically BWA saying where it thinks each sequence might align to. The next step is running them as paired end, and it's going to take the paired end data and figure out where they, based on paired end mates, where stuff belongs. So again, each of these should take only about 45 seconds each. All right. Anybody gotten both paired ends aligned? Okay, got a couple people. Who's waiting on paired end 2 alignment? Okay, so everybody should be done in just a couple seconds then. All right, I think everybody's alignment for paired end 1 and paired end 2 should be going or should be done. So the next step is running this command here. So it's going to be BWA SAMPE. So this is telling it create a SAM file from paired end data. As usual, we need to give it the reference. We're also giving it our 2 sci files, and we're going to give it our 2 sequencing inputs. And we're going to output it to aln-pe.sam, so align paired end.sam. And you should run this and you should get an error message. Are people getting error messages? Did anybody not get an error message? All right, sounds like people are still typing. I'll give you a minute to write this. It's kind of a long, awkward command. All right, sounds like the keyboards have kind of fallen silent, so I think everybody's gotten the command in. Everybody got an error message when you ran this, right? So this is my one big gripe about BWA. Unexpected end of file. Anybody have any idea what that means? BWA has some of the most unclear error messages I've ever seen. This is actually a better error message than another one I've seen, which is unable to perform psi magic. Took me a few minutes to figure out what that meant. It turns out that I had a psi file that was, I think I was accidentally zipping it at one portion, and I didn't realize it was zipped. So it looked like a bunch of garbage to the program. But they didn't say, hey, your psi file looks like it's corrupted or looks like it's garbage. It just said unable to perform psi magic. BWA has some of the worst error messages in terms of being able to fix them. However, options to get angry or remember, what did we align with? So when we aligned, we used the clean files. We were just using the original files. So if you go back and you change those original FASTQs to the clean FASTQs, it's going to take maybe 30 seconds, and you're going to have a SAM file. All right, if you're getting an error message about the index, that's not the error message you should be getting. Let's have a look. So I can tell you that you're past. Don't worry about any of that right now. If you can't find the index. I think I see the problem. So try that. Okay, and I see the other problem. Yeah, and a couple of typos in there.
Ah. Up.
Okay. Now, let me just check real quick. Let me see if I can see what's missing there. Ah, so you have you have it with the typo.
I thought I redid it too. I just put out the.
Oh, okay.
I just redid it, and so I have
Yeah.
duplicate, but.
Okay, so let's see, you have both. That's the one with the error in it.
And then the dot at the end is.
Okay, so this is what it should be doing.
Another error. I mean,
Well,
another typo.
that's, well, no, that's actually, yeah, so you had a typo in there somewhere, but it's fixed.
Okay, thanks.
Yeah. Yeah, tab autocomplete, when you start getting in the habit of using that, that'll get you around a lot of typos. So the SCI files aren't the actual alignment. The SCI files are BWA looking at each paired end individually and saying, okay, I think this read might align here, 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 or here. And then looking at paired end 2 saying, okay, this read might align in these other places. And then when you run the actual SAM creation, it's going to look at both paired end 1 and paired end 2. It's going to look at the places where they might align to. And it's going to say, okay, I have one possible alignment for paired end 1 here, one possible alignment for paired end 2 here. This is where they belong because they're in the right place relative to one another. The SCI file is kind of an intermediate step. All right. If you have single end, you're going to do the single end alignment pipeline, and the single ended alignment pipeline is very similar, but you're just doing it with you're not running it on both paired ends. You'll run the SCI step on one single end, and then you'll run the SAM step on that same thing. I'll also mention that there's a whole diversity of different aligners out there. So BWA is one of the originals, and we're basically running BWA in its original mode. What people typically do now is they more frequently run BWA MEM, which is a new mode for BWA to run in. BWA also doesn't do a good job of handling RNA data. BWA was not built to handle splicing. So if you're doing RNA, you're going to use even a different aligner. I know a lot of people used to do a top hat cufflinks-based system. Top hat cufflinks is now kind of old, and it's more or less deprecated. Now people are generally using HiSat 2 or something like that for alignment. All right, so where are we now? We just did our alignment of our raw reads to our genome. So as I said, I'm going to get everybody through the step of getting everything aligned. What do you get when you do an alignment? So if you know the head command in Linux, who here knows what the head command does? Yeah, it'll give you the first couple lines of a file. So if you run the head command on the SAM file, you'll notice that it's actually human readable. The SCI file's totally not human readable. Those look like junk, but the SAM file very easily readable. And so if we look here, we can see that we have this line that starts with an @ symbol. And this is telling us that we have chromosome 14 and what length it is. If you were aligning to the whole genome, you'd have a bunch of these lines. There's also this @PG. This is basically saying the program. And it's telling you this was aligned on BWA. And this is actually even saying what the command line that was used to run BWA was. So BWA has like this level of traceability where when it outputs a SAM file, it'll tell you the actual command that somebody used to generate that SAM file, as well as all the different versioning info and that kind of thing. Following the header lines, we have our actual data lines. So these data lines, this is all one line, but it's obviously broken up because it's a very long line of data. We can see a few things that kind of already we can tell what they are. There's this thing. This looks like some kind of an identifier, probably like the first field in our FASTQ data. This 99, don't know what that is yet. CHR14, anybody want to hazard a guess what CHR14 means? Chromosome 14. Anybody want to guess what chromosome 14 means there? What's the chromosome 14 telling you? Well, not the reference genome. Specifically, what's it telling you? What chromosome that sequence aligned to? So in this case, it is the reference genome because chromosome 14 is the entire reference genome. But it's telling you which specific part of the genome it aligned to. There's also this very large integer after the chromosome. So just by the fact that it's after the chromosome and a very large integer, can anybody guess what that is? Yeah, position. Position on the chromosome. We have another integer here, much smaller, don't know what that is. We have this thing, don't know what that is, it's 101M, some kind of code. We have an equal sign here, that's interesting. And we have another very large integer that looks actually pretty close to this one. Anybody want to hazard a guess what that very large integer might be? So not where it ends, but that's a very good guess. It actually means something else, and we'll see what that means in a bit. But you are right that it is a position. Your file may not look exactly the same. Oh, yeah, you're going to have a different file name. Yeah, you will have a different file name. So we're about to get to that. So we have another integer here, the number 170. Anybody want to guess what that might be? Yeah, length of the read. We have a string here of C's, A's, T's, and G's. Anybody want to hazard a guess? The actual sequence itself. Followed by another string of equal length that is a bunch of weird characters. What does that look like? That's your quality score. And then over here we have a bunch of values. These, I don't even know what most of these mean, and we're not going to worry too much about them. All right, so what do these lines actually mean? So we kind of guessed this looked like the first, this being as it's the first element, and also being as it looks like the beginning of a FASTQ line, we guess that this is the read name, and that is the read identifier. 99, that's called the flag score. That still tells you absolutely nothing about what it means. And I'm going to go into what it means now. So I'm guessing you're probably the only person familiar with kind of representing a bunch of true falses as an integer, basically using binary. That's what the flag score is doing. Flag score represents a bunch of true false values related to your read. So for example, a flag value of 66, we can explain that as 64 plus 2. That's telling you that read is first in pair and read is mapped in a proper pair. So by convention, if something is true, then it's basically 1, or it's added to. You'll kind of see more about this later, but I find the numbers I can add up to equal the flag value, so a flag value of 66. How do I get to 66 with these numbers? Yeah, I can only get there by adding 64 and 2, right? So that tells me that I have a flag value of 66. And a flag value of 66, since the numbers that I have to add to get there are 64 and 2, that tells me the read is the first in pair, and the read is mapped in a proper pair. What would happen? By the way, this flag value is actually one that you would probably not see ever existing. What would it mean if I had a flag value of 67? It would mean read is first in pair, read is mapped in a proper pair, and read is paired. So there's certain combinations that you're always going to find together. You should not actually find a 66 value, because if the read is first in pair and read is mapped in a proper pair, that read is paired. Likewise, you shouldn't find 4 combined with some other things, like you can't have a read be unmapped and mapped in a proper pair at the same time. So you should never find 4 and 2 going in the same value. If you ever find that you need it, there is a site where you can either check these boxes and have it explain what the flag score would be, or you can punch in the flag score and it'll check the boxes telling you which attributes apply to your read. So again, just to give an idea of how it works, each of these values, which can be either true or false, gets some power of 2 assigned to it, and if it's true, you just add that power of 2. So how does this work in terms of counting in binary? We count in decimal. Humans like to count in decimal. Anybody have a guess as to why? We got 10 fingers. So for us, if you see 1 0 written on a page, they mean 10. That means 10 to the first power. In binary, if you see 1 0 written on a page, since computers can only count to 2, or computers can actually only count to 1, a 1 0 on a page actually means 2. So in our decimal system, the first digit is 10 to the 0 power. The second digit is 10 to the first power, 10 to the second power, and so on and so on. You learn this in elementary school just as the ones, the tens, and the hundreds. In binary, it goes the 1s, the 2s, the 4s, the 8s, the 16s, the 32s, and all that. And each of these correlates back to both a numerical value and to some descriptor. So for example, here you're looking at 64 plus 32 plus 16 plus 2 plus 1. And if you add those together, you'll get the flag score for a read being first in pair, a read having its mate on the reverse strand, the read itself being on the reverse strand, the read mapping in proper pair, and the read being paired. This is a combination, by the way, you would never see in real life. The reason for that being that if the mate is mapped on the reverse strand and the read is mapped on the reverse strand, then the reads are not mapped in a proper pair, because the reads should be on opposite strands. If both reads are mapping to the same strand, that actually suggests that you had a chromosomal inversion. Somewhere between the reads, a chunk of you have one read reading in from one side, one read reading in from the other side on the opposite strand. If they both map to the same strand, then somewhere in between where those reads started, there was a chunk of chromosome that was basically taken out, flipped over, and put back in. All right, so SAM file tells you, basically it's useful for filtering. So if you want to get rid of all your unmapped reads, you can just filter on the read was mapped value, and you can say get rid of any reads where the read was mapped value is false. So your BAM file is going to include unmapped reads. It says here you'll want to remove these. I always say maybe you want to remove them. You probably don't always want to remove these. Sometimes your unmapped reads have some interesting information in them. There's some people who are writing protocols to take the unmapped reads and try and still attribute them. Sometimes your unmapped reads will tell you things that your mapped reads won't, like maybe you had organisms present you didn't know about. All right, so We pretty much guessed that CHR14 means the chromosome it mapped to. That very large integer following it immediately is the position. 60, that's the mapping quality score. That's in Fred format, just like before. So with individual reads, we said that Fred scores typically go up to 40. This goes up to 60. So a Fred score of 40 is 1 in 10,000 chance that it was a bad base call. So a Fred score of 50, 1 in 100,000. Fred score of 60, 1 in a million. So it's saying 1 in a million chance that this read actually should align somewhere else. So very high confidence this read is where it belongs. We have this thing here that's called a cigar string. Cigar string just describes how this thing aligns to the genome. So, for example, here you see 80M, 3I, 9M. That's saying 80 matches, 3 insertions, 9 more matches. So this thing aligned if you say there's a 3 base insertion in it. All right, the next two, the mate reference chromosome and the mate reference position. And as you notice here, the mate reference chromosome is an equal sign. So the equal sign is shorthand in a SAM file for mate mapped to the same chromosome, because mates generally should map to the same chromosome. And by using an equal sign, we're saving ourselves basically 4 characters per line per read, which if you have 50 million reads, that 4 characters per read actually adds up to a lot. Also, if you see a lot of situations where your read and its mate are aligning to different chromosomes, that should be a cause for concern. You should not see that happening. All right, a couple of other things. Template length for the alignment, the actual base is called, base quality, so your quality string, and then these are a bunch of just different tags that come along with the program. Yes? Oh, yes. I believe this means that it's just, it's relative to the start position. And I think you get negative template length if you're on a reverse strand, because the start position is going to go actually here and then it's going to go backwards. So I'm pretty sure that's I believe that's where you get negative template lengths from. All right. This is usually where I would go over things like sed and awk, but we're pretty much at the end of class, and Sergey actually tends to cover this more in depth than I do. But these are Linux programs for analyzing data. So I think we're pretty much done here. Everybody is free to go. I'll stick around for a few minutes and answer any questions anybody might have. Otherwise, feel free to get in contact with me. I think my email's on the website. Also, feel free to find me. I'm on ResearchGate. I'm on LinkedIn, all those places. Definitely keep in touch. I always like to see what my students are up to.